Key terms for Chapter 2. Acid, compound that releases hydrogen ions in solution. Activation energy, amount of energy greater than the energy contained in the reactants, which must overcome for a reaction to proceed. Adenosine triphosphate, ATP, nucleotide containing ribose and an adenine base that is an essential in energy transfer. Amino acid, building block of proteins, characterized by an amino and carboxyl function of groups and a variable side chain. Anion, atom with a negative charge. Atom, smallest unit of an element that retains the unique properties of that element. Atomic number, number of protons in the nucleus of an atom. Base, compound that accepts hydrogen ions in solution. Bond, electrical force linking atoms. Buffer, solution containing a weak acid or a weak base that opposes wide fluctuations in the pH of body fluids. Carbohydrate, class of organic compounds built from sugars, molecules containing carbon, hydrogen, and oxygen into a 1:1 ratio. Catalyst, substance that increases the rate of a chemical reaction without itself being changed in the process. Cation, atom with a positive charge. Chemical energy, form of energy that is absorbed as chemical bonds form, stored as they are maintained, and released as they are broken. Colloid, liquid mixture in which the solute particles consist of clumps of molecules large enough to scatter light. Compound, substance composed of two or more different elements joined by chemical bonds. Concentration, number of particles within a given space. Covalent bond, chemical bond in which two atoms share electrons, thereby completing their valence shells. Decomposition reaction, type of catabolic reaction in which one or more bonds within a larger molecule are broken, resulting in the release of smaller molecules or atoms. Denaturation, change in the structure of a molecule through physical or chemical means. Deoxyribonucleic acid, DNA. Deoxyribose containing nucleotide that stores genetic information. Disaccharide, pair of carbohydrate monomers bonded by dehydration synthesis via glycosidic bond. Disulfide bond, covalent bond formed within a polypeptide between sulfide groups of sulfur-containing amino acids, for example, cysteine. Electron, subatomic particle having a negative charge and nearly no mass, found orbiting atom's nucleus. Electron shell, area of space given distance from an atom's nucleus in which electrons are grouped. Element, substance that cannot be created or broken down by ordinary chemical means. Enzyme, protein or RNA that catalyzes chemical reactions. Exchange reaction, type of chemical reaction in which bonds are both formed and broken, resulting in the transfer of components. Functional group, group of atoms linked by strong covalent bonds that tends to behave as a distinct unit in chemical reactions with other atoms. Hydrogen bond, dipole-dipole bond in which a hydrogen atom covalently bonded to an electronegative atom is weakly attracted to a second electronegative atom. Inorganic compound, substance that does not contain both carbon and hydrogen. Ion, atom with an overall positive or negative charge. Ionic bond, attraction between an anion and a cation. Isotope, one of the variations of an element in which the number of neutrons differ from each other. Kinetic energy, energy that matter possesses because of its motion. Lipid, class of nonpolar organic compounds built from hydrocarbons and distinguished by the fact that they are not soluble in water. Macromolecule, larger molecule formed by covalent bonding. Mass number, sum of the number of protons and neutrons in the nucleus of an atom. Matter, physical substance, that which occupies space and has mass. Molecule, two or more atoms covalently bonded together. Monosaccharide, monomer of carbohydrate, also known as a simple sugar. Neutron, heavy subatomic particle having no electrical charge and found in the atom's nucleus. Nucleotide, class of organic compounds composed of one or more phosphate groups, a pentose sugar, and a base. Peptide bond, covalent bond formed by dehydration synthesis between two amino acids. Periodic table of elements, arrangement of the elements in a table according to their atomic number. pH, negative logarithm in the hydrogen ion concentration of a solution. Phospholipid, a lipid compound in which a phosphate group is combined with a diglyceride. Phosphorylation, addition of one or more phosphate groups into an organic compound. Polar molecule, molecule with regions that have opposite charges resulting from uneven numbers of electrons in the nuclei of atoms participating in the covalent bond. Polysaccharide, compound consisting of more than two carbohydrate monomers bonded by a dehydration synthesis via glycosidic bonds. Potential energy, stored energy matter possesses because of the positioning or structure of its components. Product, one or more substances produced by a chemical reaction. Prostaglandin, lipid compound derived from fatty acid chains and important in regulating several body processes. Protein, class of organic compounds that are composed of many amino acids linked together by a peptide bond substance that contains both carbon and hydrogen electrons in the nuclei of the atoms participating in the covalent bond. Proton, heavy subatomic particle having a positive charge and found in the atom's nucleus. Purine, nitrogen containing base with a double ring structure, adenine and guanine.